Humans have specialized immune cells that evolved to mount large-scale attacks on invaders like parasites, bacteria, and viruses. We call them white blood cells. A specialized form of these cells, B lymphocytes, produce swarms of antibodies to attach to invaders, attempt to disable them, and signal the appropriate destruction method. Antibodies are small, Y-shaped proteins with two important regions: the arms that attach to the invader, and the stem. The arms contain randomized regions to increase the chances they might bind new and constantly evolving invaders. The stem of an antibody functions as a signal to the immune system. When B cells are first created, they can only produce basic IgM antibodies. However, we have evolved a method to switch the stem of this basic antibody to create 4 additional classes of antibodies to target invaders with different immune responses. This location in the genome contains the instructions for building an antibody. The VDJ region encodes the arms, the C regions encode the different stems, and the switch regions between them serve as separators. At first, only the VDJ and first C regions are expressed, creating a basic IgM antibody. If the B cell is chosen to create a specialized antibody, IgA for example, the DNA between the switch regions must be removed. How does this physically happen? It all begins with a protein called activation-induced cytidine deaminase, AID. AID binds to the switch regions and deaminates a C residue. This creates a mismatch that is processed into a double-strand break by other proteins. The DNA between the breaks is looped out and discarded. Proteins Q70 and Q80 bind the ends of the broken switch regions. These ends are sensed, resected, and brought together by a team of other proteins. Finally, the gap is sealed and the break is repaired. This end repair and joining process, called non-homologous end joining, is not error-free, but can join the switch regions fast and efficiently. By sequencing these recombined switch regions and comparing the results with biochemical assays, the Pan-Hammarstrom lab has revealed new information about class switch recombination and non-homologous end joining. Thanks to class switch recombination, this B lymphocyte cell now creates IgA antibodies designed to bind the invading poliovirus and begin its destruction.